Pleasure. Okay, I'm just going to share my screen, and hopefully you can see it. Everyone's got my screen, I hope, and yeah, so it's a pleasure to be here. Thank you for the introduction, Kaiser. I have to say I've never given a seminar in my socks before, but there's a first time for everything. So before I start, if you would like to, as it's a Zoom meeting, if you go on to active speaker mode, then you can put my ugly mug in the top right in the blue box, and then I basically promise not to put any data there. So then you'll be safe. So yeah, I'm going to talk about the HUSH complex and what we've learned of its mechanism. And as a sort of stories to go with your lunch, I'm going to start by talking about the discovery of the HUSH complex and what we know of its components and organization. And then I'm going to tell two stories, more detailed stories about two of its components. One is called Taser and one is called MORK2, and how we've used structural biology in particular to learn more about its function. So throughout this talk, I'll be showing some protein structures, and of course these wouldn't have been possible without the support of various large-scale facilities. Some of them are listed here. So apart from posing an obvious threat to human health, which we all know about, especially over the last year, viruses are really all around us, and we've been living and evolving alongside viruses since the very beginning. I was struck by this remarkable statistic that nearly 1 billion viruses land on a square meter of our planet every single day. And that's really not supposed to scare you in the middle of a pandemic, but rather to emphasize that viruses are ubiquitous. And perhaps it'll help to think back to our sort of school book definition of the structure of a virus, which illustrates that at their core, really, viruses are kind of mobile genetic elements. That is a piece of genetic material, DNA or RNA, surrounded by a shell of protein. So as you probably know, some viruses can integrate that genetic information into ours or the host. And probably the most famous example of this is a human pathogen called HIV. And this illustration shows how this process happens. So the viral DNA in blue inserts itself through a biochemical process called integration. And importantly, if this integration event happens in a germline cell, the insertion of the viral genetic information can then be fixed because it can be passed on through generation to generation and therefore fixed in a human population. And now, if that happens, then the exogenous virus becomes endogenized and has become part of us, a so-called endogenous retrovirus. And it turns out this has happened a lot. So if we think about what our human DNA is made from, it might surprise you, and it certainly surprised me, to learn that only 1.5% of our genome actually encodes protein. But up to 8% of our genome is composed of these endogenized retroviral sequences, or ERVs. And ERVs are, as I described for viruses, they're mobile genetic elements, but they're now within our genome. And mobile genetic elements that are within our genome are also called transposons. So transposons can be really thought of as like the viral elements within us. And they're really defined as bits of DNA that can jump around to different targets. They can jump around DNA, much like viruses. And transposons were really discovered by a pioneering scientist called Barbara McClintock, who won the Nobel Prize in 1983. And she discovered that transposons have a lot of activity in corn and actually drive this beautiful diversity of the corn kernel color due to these germline integrations, which which drive this variation in the population. But it turns out that there are lots of these in our genomes, and these are not just the endogenous retroviral sequences. So if we go back to our human genome, it's thought that more than 44% of our genome is actually made up of these jumping genes, these transposons. So that brings me to sort of say that our genomes are really under constant threat from viral-like sequences and these transposons. And my research is really aimed at understanding how we really deal with this. So this project started back in 2015, as Kajsa introduced, in the lab of Paul Lehner. And Paul's lab did a beautiful genetic screen looking for factors, human factors, that are involved in silencing these viral elements in human cells. So they took human cells and transduced these cells with a GFP-tagged viral construct and observed a range of GFP expressions. So some cells were dim, some cells were bright. And Paul's lab then sorted out the dim population. So those cells in which the viral sequence had somehow been silenced. They then subjected this pool of dim cells to mutagenesis, either through a gene trap or CRISPR approaches, and then collected the pool of mutagenized cells in which some of the cells had become GFP bright again. By sorting these bright cells and then sequencing the mutant cells, they characterized and discovered a new complex of cells, proteins, which they called the human silencing hub, or HUSH complex. So HUSH is composed of 3 core constituents: paraffilin, Taser, and MPP8. And Taser was completely uncharacterized at the time, so they got to name it transgene activation suppressor. They also found SETDB1, which is a histone methyltransferase depositing a repressive epigenetic modification, which is known as H3K9 trimethylation, and a chromatin remodeling ATPase called MORK2. And since this discovery was made, HUSH has really emerged as a really central player in defending the human genome against mobile genetic elements in general. So we and others have discovered that HUSH regulates a subset of these transposons that I was describing before. Other labs have shown it's an important factor in mediating the silencing of HIV virus. And other labs have shown that actually it can work also on unintegrated viral sequences. The basic model behind how HUSH works is that HUSH, the 3 core components upon viral or transposon integration, somehow assemble at the chromatin where the virus has landed and recruit the MORK2 ATPase and SETDB1 to remodel chromatin. So they somehow recruit SETDB1, which deposits this repressive epigenetic modification, H3K9 trimethylation. And it should be noted that actually MPP8 itself has a binding a domain which binds this modification, illustrating how we can a sort of positive feedback loop in which the epigenetic modification recruits more HUSH complex. And HUSH also recruits an ATPase called MORK2, which is thought to be a chromatin remodeler. And this somehow shuts down the replication of this viral or transposon sequence. But as sort of biochemists and structural biologists, we looked at this model and were left with a lot of questions about how HUSH actually works. So just a few of these are: how does this complex assemble on chromatin? How does HUSH recognize its targets and deposit this repressive epigenetic mark? And how does MORK2 remodel chromatin? So my first story is about Taser, which is the central component of the HUSH complex, which, as I mentioned before, is or was a completely uncharacterized protein. So Taser is a large multi-domain protein, and Taser acts as the core scaffold of the HUSH complex. So in pull-down assays, we can show that Taser recruits the other components. It was annotated with an N-terminal domain of unknown function, but very little else was annotated at the start of this project. It then has a domain which we've shown recruits the MPP8 protein, and we're working on a structure of the complex between Taser and MPP8. And then it has another short sequence which recruits the third member of the complex called paraphilin, which I won't talk about today, but we've solved this structure and published this work this year. And of course, we can try to reconstitute the HUSH complex in various expression systems, and we've managed to do that. And of course, this will be an excellent target for cryo-EM structure determination. So I want to introduce an important assay, which will be kind of, which we'll come back to in the course of these results. And that's to use that GFP reporter that I described at the start in the forward genetic screen to give a convenient readout of whether HUSH is functional or not. So this is based on facts. So here we have a GFP reporter. You can see the HUSH repressed clone in gray, which upon Taser knockout becomes bright, as you would expect. But then if you exogenously provide the cells with Taser, so wild type in this case on the left, then you can restore the reporter repression, this epigenetic repression. But in the case of a domain deletion, for example, here, we've deleted this domain of unknown function at the N-terminus of Taser, you see no rescue of the reporter repression. And that tells us that this domain is essential for viral silencing. So we were really interested to solve the structure of this domain, especially because other studies have shown that a mouse mutation in this domain causes embryonic lethality at an early stage of development. Unfortunately, Taser, this domain of unknown function did not crystallize in any of more than 2,000 conditions that we tested at the LMB. So we turned to NMR, which illustrated a folded domain, but with evidence of flexibility. So if you're not familiar with an NMR HSQC spectrum, each peak is then assigned to one residue in the protein, and the peak position and also intensity gives a measure of its chemical environment, but also its dynamics. And this analysis illustrated that we had a pronounced loop region which had high levels of disorder right in the middle of the domain. So we went ahead and deleted this most disordered internal loop. We checked that it didn't cause a change in HUSH function, and it didn't, but it enabled us to grow beautiful single crystals of this domain. And we solved the structure of this domain to 2 angstroms, which is shown in green here. And then this was at the Diamond synchrotron in Oxford. And we found that this domain of unknown function closely resembles a PARP catalytic domain. So shown is the PARP1 catalytic domain, which is kind of like the canonical member of the family. So what do PARPs do? PARPs usually add NAD-derived chains of poly-ADP ribose onto nuclear proteins in response to DNA damage. And this was interesting to us. So usually this domain works through a catalytic triad, this histidine, tyrosine, and glutamate. And in response to binding a double-stranded break, it uses NAD to add these chains, these negatively charged chains of poly-ADP ribose to its substrates. And this alters protein-protein interactions, but also accounts for chromatin remodelling. So we were really excited to see a PARP domain in Taser. But what we found was that Taser is actually not an active enzyme. So we've described Taser as like a pseudo PARP. And zooming into its active site, and then comparing with the PARP1 active site, you can see why this is. So here we have in red the PARP1 active site bound to a mimic of its cofactor NAD. But in Taser, we have a loop that closes over the active site. We have a tyrosine that actually plugs the gap, but also we have mutations across the active site, which remove this key catalytic triad and the active site becomes highly hydrophobic. And therefore, we weren't surprised to see that when we measured binding of a PARP inhibitor, whereas PARP1 binds in a concentration-dependent manner, we saw no binding with Taser. But one thing we observed during purification and during our analyses was that this domain has lost its catalytic activity, but acquired a new RNA binding function. So this is the structure again, shown in a slightly different way. And we observed that although it's lost its active site, it's acquired these new loops, not the flexible loop that we mentioned before, but a different loop. And this loop seems to be important in interacting with RNA. So we were able to measure this and also find a mutation which did not disturb the fold of the protein, but impaired this RNA binding and crucially destroyed HUSH function in silencing. And what we were really amazed to see was that a single point mutation in this Taser pseudoPARP domain is actually sufficient to wipe out HUSH function genome-wide. So here we're going to look at this H3K9 trimethylation, which is this repressive epigenetic mark. And this is the control where we have on the left a stacked up version of all of the target sites for HUSH. And then when we knock out Taser, we lose this H3K9 trimethylation mark. If we bring back the wild type, you see a restoration of this, this mark. But in this point mutant, we've completely wiped out function. So we've basically got a point mutant which replicates the complete knockout. So some thoughts from this first story. HUSH is a newly discovered epigenetic silencing complex. It spreads H3K9 trimethylation to protect the genome against integrating viruses and transposons. And we think we've identified many of the key molecular features of Taser, but we've also identified a new RNA binding by this pseudo-PARP domain in Taser. And think that perhaps this has a role in sensing the nascent viral transcripts and recruiting HUSH to chromatin. So story 2 is about a protein called MORK2, and MORK2 is an essential effector of HUSH, as depicted here. If you wipe out MORK2, this GFP viral construct becomes bright again. So the Mork family, there was a bit more known about compared to what we started with Taser. And Morks are known to be ATPases, which are responsible for transgene silencing in many walks of life, including nematodes, plants, and mammals. And since I made this slide, they've even been found in parasites. So in terms of the mammalian Morks, we've got 4 Mork proteins in our genomes. Morks 1 to 4. They all have an N-terminal GHKL type ATPase module, shown in yellow. They all have this CW domain, which is a zinc finger which binds histones. And they all have a C-terminal coiled coil, which enables constitutive dimerizations through the C-terminus. But just focusing in on MORK2, which is our factor of interest, we asked, how does MORK2 work as a chromatin remodeler? Very little was known about MORK2. So this time at ESRF, we solved the structure of the ATPase module of the MORK2 homodimer in complex with an ATP analog. And you can see it forms this beautifully symmetric M-shaped molecule, M for Mork, where the ATPase module is interrupted by a coiled coil insertion shown in green, which presents these sort of projectile arms coming out of the core of the protein. We've also solved an NMR structure of one of the chromatin binding domains, which I won't talk about today. And of course, putting this all together, this is an excellent cryo-EM target, and this is a cryo-EM micrograph that we collected at some of the microscopes in Diamond in Oxford. And we're working up towards a model of the entire structure. So what the structure showed was that ATP binding actually induces a MORK2 dimer interface, and this structural switch is really critical for silencing by HUSH. So we found when, and this is kind of our model of how it's working, the C-terminal coiled domain is constitutively dimeric. So upon ATP binding, this ATPase module at the N-terminus becomes a dimer. And what we found was if we mutated an ATP binding site, which is this N39A mutation, or we mutated the dimer interface itself, but without removing ATPase function, we lost the capacity for HUSH to silence. So this dimerization of the N-terminus upon ATP binding is really critical for the function of HUSH. We also observed that these projections, these unique coil-coil insertions into the Mork structure are highly positively charged. And we asked the question of whether this positively charged arms have a role in binding to DNA. And we found that several charge reversal mutations at the tip of this arm caused complete loss again of HUSH function. And we could show in vitro that binding to nucleosome core particles became substantially weaker in the presence of these charge reversal mutations. So in other words, the N-terminal dimerization of MORK2 can bring together potentially two DNA duplexes, which we think is a nice mechanism for how this ATP-dependent structural switch is crucial for HUSH silencing. One of the really interesting things about this protein as well was that it had been identified over the past few years that mutations in this ATPase site cause a range of neurodevelopmental disorders. So a number of clinical reports were published while we were doing this structural work. And so we were able to then look on our structure at how this may affect both MORK2 dimerization and ATPase activity, but also then look in our functional assays at how HUSH was affected. What we observed was a range of effects on ATPase activity, but to our surprise, the variants that gave lower ATPase activity actually led to better silencing of the GFP reporter. And we were able to resolve structures of some of these mutants to understand why, but I won't go into the details of how those work now. But in other words, what we observed was as ATP hydrolysis rate increased, HUSH silencing actually decreased, which was kind of the opposite to what you might expect. But so we put together a model of how this might work, where we know that the N-terminal monomers bind DNA, and that dimerization through ATP binding would lead to bringing the two duplexes together. And so conceptually, if you decrease the rate of ATP hydrolysis, you kind of shift the equilibrium towards the right. And we think the right-hand situation where you have the N-terminal dimers formed is kind of like the HUSH silence state. So some more like take-home messages or stay-at-home messages. So those are the two messages that I had before. We've also shown that MORK2 key HUSH effector forms these ATP-dependent clamps around DNA, and we've illustrated that this is an essential process in HUSH chromatin regulation. And we've also shown that patient mutations in MORK2 misregulate HUSH function, and we think this is due to somehow perturbing the dimerization dynamics of the protein. and this ultimately leads to disease. And our model is just shown here. We think we've identified a key RNA binding activity of HUSH, but also started to understand how MORK2 is recruited and works as a remodeler within the complex to silence the underlying viral or transposon DNA. And with that, I'd just like to thank colleagues in Cambridge with whom I worked for much of this project in the lab of Yorgo Modis and Paul Lehner. In particular, thanks to Richard Timms and Eva Czasownikarova, who really pioneered this forward genetic screen to discover HUSH. I'd like to thank BBSRC, which is a UK funding agency, for funding me for 3 years when I was doing this work. I'd like to thank SSMF, who's funded me to come to Lund. And thanks in Lund to Johan Jakobsen, who's my host at the moment while I establish a team. So just to say briefly, I'm a new PI at the Lund Department of Medicine. And I have this SSMF funded program on the mechanism of chromatin regulation by Mork ATPases. And I'm planning to hire a postdoc to start later this year to try to understand the role of transposon silencing in Mork associated neurodevelopmental disorders. And with that, I think that's 25 minutes. So I'd be really happy to take questions. Or if you can't think of questions, but something comes to you later, very happy for you to email me or get in touch if you find any of this interesting, just want to discuss HUSH and chromatin and transposons. I'd be happy to do that anytime. So thank you very much. And I'll stop, well, maybe, yeah, I'll stop sharing now.
Thank you very much, Christopher. It was a very clear and interesting talk.